Apex predators are important globally. They monitor and regulate biosystems throughout the world and are very important in times of turbulence and times of change. Perhaps the influence of the apex predators in Yellowstone National Park is one particular example of how important those wolves are to maintaining biodiversity in Yellowstone National Park. In Australia, the apex predator is the dingo. The Australian dingo arrived on the continent somewhere between 5,000 and 8,000 years ago, likely through Southeast Asia. The Australian dingo probably has at least two types that we are currently calling ecotypes. One is the desert ecotype and the other is the alpine ecotype. These alpine and desert ecotypes have traditionally been described based on cranial structures, which are useful but have problems associated with them if you want to try and identify a living animal. The Australian dingo is very important in Australian society and are totems of First Nation Australians. They're also important in maintaining biodiversity in multiple Australian ecosystems throughout Australia. However, there is debate about the role of the dingo in Australia because there is strong evidence of hybridisation between dingoes and domestic dogs. This hybridisation creates an animal, a wild dog, that potentially has different behaviours from the ancestral dingo type. One of the things that we are interested in is distinguishing between the true dingo and the influence of wild dogs in the Australian ecosystem. I'm sitting here in Pure Dingo Sanctuary. So, Barry, when did you find Sandy and what were the circumstances around that finding?
Yeah, g'day. My name's Barry and that's my wife Lynn. And we were doing one of our frequent trips to the outback. In this particular case, near the Simpson Desert, the Strzelecki track. We were fortunate enough to find 3 of these little pup desert dingoes. They were about 3 weeks old at the time. They were in very poor shape. They were infested with sarcoptic mange mites and they were probably pretty badly dehydrated. It was early summer. We then made the decision that we would try to save them, which we did, and then we made arrangements to travel home because it was not possible to take them with us at that particular time. So we returned home to the central coast of New South Wales, and then within a day we turned around and went back with the intent to pick them up and bring them back with us.
When the animals, the canids, came back with you to central coastal New South Wales, you then had some blood drawn and you sent that blood to me for genetic testing. What was the result of those tests?
When we got the results back from you regarding these blood tests for these dingoes, it was a very pleasant surprise to find out that they were pure dingo. I was suspicious that they had dingo in them, but I had no idea that they were pure dingoes. I mean, they could have been dumped out there. But when we found out that they were pure dingoes, it was even more important for me to try and look after them and maintain their health and take them under our wing and do what we could and liaise with Bill regarding...
So the next step in this story is the announcement in 2017 of the Pacific Biosciences World's Most Interesting Genome Competition.
Well, Sandy was announced as the winner of the desert dingo being the world's most interesting genome in 2017. This enabled us to get a high molecular weight sample taken from Sandy The DNA was shipped to Dave Caderna's lab at Arizona, and he used 31 sequel-2 cells to sequence the genome of Sandy to about 60x coverage. These samples were then sent to Computomics in Germany, where Falcon was used to construct a de novo assembly of the Sandy genome. Because we didn't have a chromosome level assembly, we decided not to publish the data, but to continue collecting data. We were able to construct version 2 of the Desert Dingo genome. So for this Desert Dingo genome, we used the 31 SMRT cells that were originally collected for version 1 of Sandy. We added Prometheome data, Hi-C data, chromium data, and some bisulfite sequencing. We compared this genome with CanFam GSD, which is the German Shepherd dog, which has a very similar array of protocols that we used to construct the de novo assembly, but there were 18 SMRT cells. So if we look at some of the information from the Desert Dingo version 2, we can see that, like the other assemblies, it has a total sequence length of about 2.4 gigabases. Pleasingly, the number of contigs went down. There were 228 contigs with a contig N50 of 41 and a contig L50 of 20. The number of scaffolds was 159, with a number of gaps being 69. This is considerably less than the number of scaffolds than CanFam. The quality of these assemblies enabled us to determine the number of Amylase copies. This was important to us because Amylase copy number has been shown to be duplicated in all domestic dogs. In the Desert Dingo, there was 1 copy of Amylase, and there was no evidence around this region of any loss of Amylase copies. So it has the ancestral copy number of 1, which is the same as wolves. A couple of very interesting points that I'd like to discuss here. There's chromosome rearrangement on chromosome 16 of Sandy on the left compared to Tasha and Nala, the boxer and German Shepherd, respectively. At this stage, we do not know whether this is particular to Sandy. We do not know if it's particular to Desert Dingoes or dingoes themselves. So it's going to be something that we need to investigate further. In terms of the methylation patterns, there were 7 gene promoters showed differences in methylation between the dingo and the German Shepherd. One of these was particularly interesting to us, which was GAL3ST1, because that was associated with galactose metabolism and provided a link with amylase 2B. We looked at that in a total of 8 dingoes and 8 German Shepherds. We looked at the expression patterns, and as predicted, methylation caused a reduction in the expression of GAL3ST1 in the German Shepherd. So for those not so familiar with amylase, wolves and dingoes have shown a single copy of amylase, adaptations allowing dogs to thrive on a starch-rich diet, including a significant AMY2B copy number gain, constituted a crucial step in the evolution of the dog from the wolf. It's thought that this probably occurred somewhere around the Neolithic, when people started to cultivate rice. They then fed tamed and then domestic dogs rice, and it was those animals, those canids, that increased in fitness due to that feeding, that were more likely to be associated with humans at that time. Based on that result, we decided to construct an experimental study. We used 2 dingo sanctuaries and the 2 German Shepherds kennels. These were all within about a 100-kilometre radius, so the environments were very similar. What we did was that we fed them an antibiotic that was to reduce the microbial diversity. We then fed them probiotics over the next 2 days to colonise their microbial systems. with constant microbial diversity. We then fed them for 10 days with a constant food and constant water. We then fed them for 4 days with increasing rice in the diet. This was aimed to stress amylase and to see whether there was going to be an increase in the response of amylase. At the beginning of the study, we also took soil and scat. We did not see any differences in the microbial communities in the soil or the scat at the beginning of the study. So this was quite a large study. This was the group that was involved at the Bargo Dingo Sanctuary. There was about 10 to 12 people that were involved in changing the food of the dingoes. There were also, of course, the veterinarians and the veterinarian's assistant that was necessary to come out to each of these to draw blood and collect scat. So this is a picture inside the Bargo Dingo Sanctuary Visitor Centre, and it was just 2 months after this time that the fire swept through the Bargo Dingo Sanctuary, and this is the same visitor centre 2 months after. It was in December 2019, and as you can see, the visitor centre is completely destroyed. Fortunately, we were able to get all the dingoes out before the fire came through. So 3 things that I wanted to illustrate from that study. I want to talk a little bit about cholesterol, because high LDL concentrations in humans has posed an evolutionary advantage by increasing the ability to fight infection through increasing the proportion of inflammatory macrophages. So the prediction was that dingoes would have higher cholesterol and higher LDL, and that would therefore give them an increased ability to fight infections in nature rather than domestic dog, which has a more sanitized environment. We then wanted to look at the Scat microbiome. We're going to look at, we looked at the total diversity in dingoes and German shepherds, and we also looked at enrichment of families linked with starch metabolism. Finally, I'm going to talk a little bit about the Scat metabolome. The major reason we were interested in that is that can we make progress towards excluding dingoes from farmlands by developing a chemical fence? In Australia, farmers shoot a variety of different wild dogs and dingoes, and they hang them up on their fences. This is a devastating picture to illustrate the problems that they have with dingoes and wild dogs. Of course, the farmer doesn't know whether these animals are pure dingoes or dingo dog hybrids. Perhaps developing a chemical fence would enable the pastoralists and the native ecosystems to exist in a little bit of harmony. As expected, dingoes had higher cholesterol than German shepherds, and this was highly significant. We then looked at cholesterol fractions in VLDL, LDL, and HDL. Notably, it is only the component, which is LDL, which differs between the dingo and the German shepherd. This supports the hypothesis that dingoes are more likely to be resistant to infection than domestic dogs. We then wanted to look at the microbiome, and the initial analysis was just a comparison of the alpha diversity. Shannon's index shows the same trend. The analysis of the microbial composition showed that one microbial phylum, 17 families and 51 genera differed between the canids, with German shepherds having a significantly greater alpha diversity than the dingoes. In terms of enrichment, the family Clostrididae and the genus Clostridium sensu stricto were enriched in dingoes. It's the only family that was actually enriched in dingoes. All other families were higher in German shepherds. Clostridium sensu stricto can utilize complex resistant starch that will have not been broken down by the dingoes due to the low amylase activity. So these are the 4 that are particularly relevant to the amylase and to starch digestion. Potentially this may provide an indicator that the pure dingo and the wild dog are actually have different food preferences. So this is another area that is going to be a spin-off of this study that we would like to explore. As far as the Scat microbiome is concerned, there was one chemical that was higher in the dingo versus the dog. Phenylethyl alcohol was higher in the dingo The only other two chemicals that differed between the scat of a dingo and a German Shepherd were ethanol, 1-phenol, and quinolone, and these were significantly higher in the German Shepherd. It's interesting that the chemists amongst us described phenylethyl alcohol as being similar to the smell of an old rose. Whether we can use the differences in proportions between these chemicals to create something like a chemical fence. In Australia, there is a fence that's called the Dingo Proof Fence that runs for over 3,000 kilometres. It's the largest fence in the world. And on one side, there's dingoes, and on the other side, there's pastoralists. This fence has partial success. If we could replace this fence with a chemical barrier, I think that would be a tremendous advantage, because it would enable other wildlife to move across this barrier and wouldn't exclude those other native animals, such as emus and small marsupials, from colonising the different regions. So in conclusion, our study underpins the dingo's genomic and ecological distinctions from purebred dogs. Our genome assembly has high contiguity. We found unique inversions in the dingo indicating differential evolutionary signature. The epigenetics analyses indicated 7 genes that are differentially methylated in the dingo compared to the German Shepherd dog. And the organismal studies provide insight into the distinct physiology of the dingoes and suggest they have a heightened immune responses, a distinct microbial community, and of course, differences in their scat metabolome that can be utilised for future research and future studies. So the future studies are going to be to examine the link between the dingo genome and ecology and physiology. That's very important in Australia, because wild dogs are considered to be Australia's biggest agricultural problem and cause a significant amount of damage throughout Australia. We're interested in quantifying the genetic variation within dingoes. We need to include some fossil data in that, and we're working with some researchers at the University of Adelaide to include some fossil data. We're interested in determining the phylogenomic position of dingoes, where we have completed the alpine dingo genome. This is the alpine dingo that we've sequenced. The sequence is available on DNA Zoo. This is Coinda. We've sequenced in collaboration with Wes Warren, Basenji. We've just finished sequencing 2 domestic dogs. One is the cattle dog, which is thought to be derived from the dingo. And we've also sequenced a chow chow. That sequence was done by DNA Link in Korea and produced some excellent hi-fi data that we were able to assemble and to get high quality resequencing assemblies. We've also started sequencing in collaboration with Tim Smith in Nebraska, African wild dogs, and this will be the outgroup to our study, as we understand that a number of groups are sequencing wolves as well as coyotes. A variety of different co-authors, 25 co-authors were involved in this study. I'd like to thank them all, those from Australia, Europe and the US. Particular thanks to the sanctuaries and kennels. There were a number of different centres that were involved, funding the all-important funding. We'd like to thank all the PacBio voters and for Emily Huttis.
So final thanks has to go to Barry and Lynn for collecting and finding the 3 amigos and bringing them back to Pure Dingo Sanctuary. And special thanks to Sandy herself, because the world now knows her both from the outside and the inside. Thanks very much, Sandy.